Hi, my name is Sammy Kishlevine, and this summer I'm interning at the Field Museum of Natural History through the museum's NSF-funded REU program. I'm studying lichens with Dr. J.P. Huang and Torsten Lumsch. The purpose of this study is to create a phylogeny that shows the evolutionary relationships between different species of Xanthoparmelia. To do this, I have to identify and compare different lichen samples using morphological, chemical, and genetic methods. I am also looking at how the environment of the lichens shapes patterns of diversity within the genus. You've probably seen lichens before, but you may not know what they are. Lichens are a composite organism made up of a symbiosis between a fungus, the mycobiont, and an algae or cyanobacteria, the photobiont. The photobiont provides nutrients for the mycobiont through photosynthesis, and the mycobiont provides UV protection for the photobiont through the use of secondary metabolites. Secondary metabolites are compounds that are not involved in reproduction or growth. They are often used as a defense against herbivory. Different species of lichens have different secondary metabolites. Because of this, we can compare the chemical profiles of different lichens in order to identify the species. The genus of lichen that I'm studying is called Xanthoparmelia. Xanthoparmelia is the biggest genus of lichens with over 800 species found worldwide. However, I'm focusing specifically on populations from Australia and Mexico. One way to tell lichens apart is to look at their physical characteristics. Botanists often use a botanical key to tell species apart. Common features used to identify lichens are the color of the upper and lower surface, the branching pattern of the rhizines, and the shape and size of the thallus. The rhizines are root-like structures that cling to the substrate, and the thallus is the leafy part of the lichen. Oftentimes, botanical keys will also use the presence or absence of a compound in order to identify the species. The method of chemical identification that I'm using is called thin layer chromatography, or TLC for short. TLC separates out the chemical compounds in the lichens on a silica plate, making them easy to visualize. The last step is the genetics work, which is the most important step in creating a phylogeny. I extract DNA from the lichens and amplify it using a method called polymerase chain reaction, or PCR. After the DNA has been amplified, I visualize it through a method called gel electrophoresis. In this photo, the different bars show the sizes of the different DNA fragments from each sample. Lastly, I sequence the DNA. The sequence of a specific region of DNA is compared across different species. The less similar the sequences are, the less related the species are. The region of DNA I am using is called the internal transcribed spacer, or ITS, region. My project is important because the correlation between environmental factors and lichen biodiversity has been previously understudied. With the information gleaned from this study, we can learn more about lichens and the mechanisms of speciation in general.